Translation going from the RNA to the protein. So the major molecules that are involved in this particular process are the tRNAs, the transfer RNAs that are getting the amino acids. One end is going to bond to the amino acid, so I'm putting A in here for short form for amino acids, and when it's bonded we call it an amino acid tRNA. The other end is going to have an anticodon that matches a particular codon, and so again this is made of RNA, so you have these complementary pairing that can happen, and so the anticodon part of it is going to match the codon part of the mRNA. There are, again, because of the wobble effect, there are not 64, because we could have 64 anticodons. There's not going to be 64 tRNAs. There's only somewhere around 40-ish of them. And this is because, again, even though they don't have the exact anticodon for a particular codon, that wobble effect, again, means that that third one's not so important because a lot of the amino acids, depending on the amino acid, that a lot of these amino acids have the same nucleotide as their third letter, and therefore whether it's one or the other doesn't matter, and therefore you can have some redundancy in the tRNAs. So this is what they look like. They're essentially a sequence of RNA. One end grabs onto the amino acid, and the other end down here, this is the anticodon. And so its letters will be complementary to the mRNA strand that they would work with. A particular codon will show up on the mRNA strand, and there will be a tRNA that matches it. Its anticodon will match the codon of the mRNA, and it will have the right amino acid attached to it to bring to the mRNA. Now, of course, the place where this is all happening is the ribosome. So this is made up of rRNA as well as some proteins. It works as the machine that actually assembles these amino acids into proteins. There's two subunits, and they are going to work by grabbing onto the mRNA, and then the tRNAs are able to bring the amino acids, and the protein is able to start building. Multiple of them can attach the same mRNA, so one can go through and make a protein, and another one can follow right along, making a second version of that same protein. Each ribosome has 4 binding sites. First one where the mRNA is attached, so it actually can read through the mRNA, and then there are 3 sites where the tRNAs are involved. There's the A site, and this is going to hold the arriving amino acids with their, arriving tRNAs with their amino acids. The P site is where we actually have the production of the amino acids, not what the P stands for, but it works. And the E site, which actually is for exit, is going to be there as well. So there's our mRNA. This blue guy here is our tRNA, and you can see it's holding on to an amino acid. So those ones we know. And then the ribosome is going to be these 2 parts here. They've sandwiched the messenger RNA so they can sort of read it. And there are 3 sites to it. So 1 is the first one. It's actually on the right. And then we have 2, and then we have 3. So they arrive, they start producing the protein, and then they exit, the tRNAs. There's the actual letters of their form. This happens in 4 steps. So there's initiation, elongation, termination, and modification, similar to our transcription. The steps themselves are very different, though. So the first one is that 5' cap that was added as the modification for the mRNAs is going to be recognized. And so we've got this beginning part to the mRNA. And so that's where the ribosome knows to bind. And by knows, I mean interacts with it in a way that causes it to bind. And somewhere in from there is going to be our start codon. The tRNA that matches, AUG being the only start codon, is going to match our anticodon of a tRNA. So we have this particular tRNA that's going to start all proteins, and its anticodon version is UAC, and it is going to be carrying this MET amino acid. The small ribosome unit is going to bind with the mRNA, and it is also going to be read from the 5' to the 3' direction. And so essentially the ribosome is going to be moving this way down the mRNA. So we have our UAC anticodon. Once that first tRNA binds, the large subunit joins in as well, and we have our functional ribosome. The start codon with its anticodon tRNA with the MET amino acid is going to be in the middle, and our A site is ready for the arrival of a new one, and E site is ready because the whole thing is going to move 1 codon to the right, which will cause this tRNA to now be in the E site, which is where it will exit. So this is the elongation part. So the new amino acid with its tRNA, so whatever the codon is here we have GUC as our second codon. So it would have a tRNA with an anticodon of CAG, and that particular one, if that's what it was, it would be valine. Now, the start one has to be the met amino acid because there is only one start codon, so that's always going to be the same. But from there on, depending on what protein, it'll just be whatever codons are needed for that particular protein. So the next codon, whatever it may be, will have its matching tmRNA land in that A site. And what will happen there is that the two amino acids, and it's a ribosome, so it's acting as a allowing this reaction to take place with the building of the peptide bond between the two amino acids. So now our met and our val are going to be covalently bonded together. We are beginning to form a polypeptide. Two is not quite poly, but you get the idea. So when this happens, so we have the bond happening, and now these guys here are a chain, or the beginning of a long chain of amino acids. We've got two so far. The ribosome is going to move over. So the whole ribosome is going to move over. And again, so what's going to happen is this first tRNA will now be in the exit spot. The one that just arrived will be in the middle spot, the P spot. And then the arrival spot is now open, ready for a new tRNA with an anticodon that matches whatever codon the ribosome just moved into. So they moved over one codon, and so what was not yet at the arrival site is now at the, I'll call it the arrival site, and the tRNA that is going to match that is going to be able to arrive there. Again, this process would repeat. So whatever amino acid came with it is going to get bonded on to the second amino acid. We'll get a peptide bond there, and then we'll have a 3 amino acid polypeptide chain starting to grow. So the whole thing keeps happening. You get the ribosome moves over, a new tRNA arrives at the A site, and then the amino acid gets bonded, polypeptide bond, to the already existing chain. Keeps happening, keeps happening, keeps happening, and eventually you get a longer and longer and longer chain until eventually there's a termination sequence. So one of the stop codons will eventually get into the A site, and when there's a stop codon in the A site, there is no matching tRNA. So what happens is, instead of a matching tRNA, that there is none, there's a release factor that basically comes in, breaks up the party. As soon as it lands, everyone leaves and goes home. Protein that is produced through this process is not yet modified. So this is, at this point, a long string of amino acids. What can happen is that it is going to be modified in several ways. So some of them are, you have this sugar added to them. They could be phosphorylated or some other chemical modifications. But we do know of certain modifications we've seen with proteins before. They will go through their primary structure, which is essentially their sequence. Then they will be modified into a functional protein. That can involve cutting or adding these other things to it, phosphates, sugars, and then the folding can begin. We have our primary structure, which then you have the hydrogen bonding that happens within that primary structure, giving you helixes or plated sheets. Then you have the R groups of those particular amino acids starting to interact with each other, charges and polarity and non-polarity. And then you could have multiple of those polypeptides coming together to form this functional protein. It may be functional already, but it may need this quaternary structure to become functional. And then you have your functional protein.